Good afternoon, everyone, and welcome to today's MECFS Stakeholder Engagement and Communication Call, which we call SEC or SEEK. My name is Christine Pearson, and I'm the Associate Director for Communications in the division where the MECFS program is located within CDC. As you may know, we host these SEC calls twice a year as part of our regular outreach and communications activities to provide information for people with CFS, as well as their loved ones, clinicians, and anyone else interested in the disease. Our goals during these calls are to provide updates on the works of CDC's MECFS program and for you to hear from external experts in the field. Today, we'll hear program updates from Dr. Elizabeth Unger, the branch chief for the chronic viral diseases branch at CDC, where MECFS is located. Then we'll turn it over to our guest speaker panel. After the presentation, we'll have a Q&A session. We have a really full schedule today, so we ask that our speakers stick to the timing as much as possible, and so that we have as much time as possible for questions. During today's Q&A, you'll have the opportunity to ask questions through the webinar platform or by phone if that's how you joined. We'll provide more information on that when we get to the Q&A session. Before we start, I'd like to remind everyone the call is open to the public, so please consider that before sharing personal information. We're also recording this call. Please disconnect now if you have any concerns with the recording. We'll post the transcript and the video as soon as possible after the call is complete. If you would like access to the closed captioning or to read along with the text of the program update, the links to both of those are posted in the chat box. Now, before we get to Dr. Unger's remarks, I'd like to share a short update on a CDC-wide initiative to improve the agency's website. As part of this project, CDC communicators and scientists are currently hard at work reviewing, updating, and reorganizing pages on cdc.gov. The goal of this is to create a more readable, user-friendly cdc.gov that meets all of our audience's needs. Now, a little bit about why our director's office created this initiative. First of all, CDC's entire website is huge, over 200,000 pages, and is extremely hard to navigate. Some pages are duplicative, others are outdated. Our research shows that web users can't find the information they need, and they struggle to find the information they're looking for. With a fresh start, we're hoping to focus on improved communication for all of our audiences, whether general public, partners, healthcare providers, or state health departments. We know that CDC's MECFS website serves a need for this community, and it's imperative to help raise awareness about MECFS and the disruption it causes in the lives of people affected by it. We're dedicated to making sure that the MECFS information is available for you, for healthcare providers, and for people who may be diagnosed in the future. Rest assured that nearly all of the MECFS current content will continue to be available on the CDC website. Some of the changes that you may anticipate or may notice are a more mobile-friendly layout, easier navigation, pages that are easier to read. In some cases, you may notice shorter sentences, shorter paragraphs, more plain language, and less jargon. Pages for healthcare providers will continue to use a higher reading level language than general public pages. In addition, some information for meetings held more than 2 years ago, and some of our older voice of the patient features will be put into an archive. This information will continue to be publicly available on an archive called the CDC Stacks and on a new archive website, which is archive.cdc.gov. This is a separate environment from the cdc.gov main site, but will be a place that you can easily access the information that we currently have. We'll be redesigning the website now through the end of March, when CDC plans to launch the new cdc.gov. We're excited about the new site and hope that it can provide benefits for all of our users. If you have questions on Clean Slate, which is the name of this initiative, you may ask them during the Q&A section of this call or by sending an email to our MECFS mailbox at mecfssec@cdc.gov. And now, before we turn it over to Dr. Unger, I'll remind you that if you have suggestions for speakers or ideas for future SEC calls, you can also send them to that same mailbox. And you could also use this address if you'd like to be added for email notifications about future calls. Now I'll turn it over to Dr. Unger to start the program.
Thank you so much, Christine. We really appreciate that you took the time to tell our audience about CDC's Clean Slate program. We hope that it will improve our online information, and we look forward to continuing to work with you and other CDC communicators on this initiative. I'd like to welcome everybody to the 22nd SEC call. To allow time for our speakers and the question and answer period, I plan to keep my remarks short, but I would like to share some brief updates on our MECFS program activities. We entered into a cooperative agreement with the CDC Foundation in December of 2022 to launch an effort called the Infection-Initiated Chronic Conditions Understanding and Engagement Project, or IQ for short. The project brings together patient partner groups and community-based organizations who are actively working to help people with MECFS, long COVID, and other infection-associated chronic illnesses, and to raise awareness of these conditions. The IQ project team is made up of members of the CDC Foundation, the Long COVID Alliance, and Commonality Incorporated. The project leaders include members of Solve ME, Dysautonomia International, the COVID-19 Long Hauler Advocacy Project, and the Patient-Led Research Collaborative. In 2023, the IQ project brought together more than 60 key partner organizations to identify and prioritize the community's needs and shares goals. The CDC Foundation worked with the Long COVID Alliance and Commonality to organize and host an IQ national webinar titled Opportunities for Action, Infection-Associated Chronic Conditions on October 24th, 2023. The webinar featured an overview of the priorities and goals identified during meetings with participating organizations this year and was attended by 375 attendees. Prompted by the increased recognition of unexplained post-acute infectious syndrome resulting from long COVID, CDC launched a new webpage in October titled Chronic Symptoms Following Infections. This webpage gives an overview of the possible causes of chronic symptoms following acute infection and how they affect people. It briefly addresses talking with your healthcare provider about your symptoms and also touches on the disease agents that have been linked to chronic symptoms. There are many similarities in the post-acute infectious syndromes and those experienced by people with MECFS. CDC's experience with MECFS guided development of this new webpage. We believe that by looking for similarities and differences between MECFS and chronic symptom syndromes that develop following a variety of known and unknown acute infections, we'll be able to learn more about how often and why they occur, as well as how to diagnose and treat them. CDC continues to partner with our collaborators on a variety of MECFS and long COVID projects. We've started sharing our findings from various studies and projects that our MECFS program supports. At the 2023 International Association for Chronic Fatigue Syndrome Myalgic Encephalomyelitis Conference, CDC and its partners collectively presented 5 abstracts and conducted 2 workshops. 2 abstracts contained data from the multi-site clinical assessment of MECFS, or MCAM, to look at how often patients had comorbidities such as dysautonomia and chronic overlapping pain conditions like fibromyalgia, irritable bowel syndrome, or chronic lower back pain, and how these comorbid conditions affected patients. We found that the NASA Lean test and Compass 31 questionnaire were useful tools to identify orthostatic intolerance in other dysautonomias. These comorbid conditions were often present and were associated with worse symptoms and function in patients with MECFS. In addition, we shared early findings from several of our long COVID projects, including an oral presentation entitled Addressing MECFS-like Symptoms in Long COVID Patients, and another presentation entitled MECFS-like Illness Among Patients at 3 U.S. post-COVID clinics, which discuss the overlap in clinical presentation between MECFS and post-COVID conditions. For surveillance activities, we continue to partner with CDC's National Center for Health Statistics to track MECFS and long COVID through the National Health Interview Survey. The September issue of NCH's Data Brief Survey provided nationally representative prevalence estimates of long COVID in adults and children. Using 2022 data from the National Health Interview Survey, we estimated that 6.9% of adults ever had long COVID and 3.4% had long COVID at the time of the interview. The prevalence rates were lower in children. 1.3% of children ever had long COVID, and 0.5% of children had long COVID at the time of interview. We also used 2021 through 2022 National Health Interview Survey to track MECFS. The data brief report was just released in the December issue of NCHS Data Brief. Based on self-reported diagnosis by a healthcare provider, we estimate that 1.3% of adults had MECFS in those years. We will continue to use this survey to track MECFS in 2023 and 2024. Now I'll turn to an update on our program's educational activities. The newest online course introduced during the last C call launched in September. It is titled MECFS Diagnosis and Management in the Age of COVID Expert Insights, and will be available for continuing education credits for 3 years. The MECFS experts who participated include Drs. Anthony Komaroff and David Systrom from Harvard Medical School, and Dr. Lucinda Bateman from the Bateman Horne Center, an MECFS specialty clinic in Salt Lake City, Utah. The course is being promoted by WebMD, the National Association of School Nurses, and other partners through their member listservs and networks. Through the 7 online Medscape courses, we reached more than 120,000 learners, had 49,000 test takers, and issued about 40,000 certificates. For a final update, I'm tasked with making the bittersweet announcement that Dr. Jennifer Cope from our Chronic Viral Diseases branch has accepted a new position and promotion within CDC. We wish her well in her new role and thank her for her time and service to CDC's MECFS program. Now I'd like to introduce Dr. Vicky Whittemore. Dr. Whittemore received a Bachelor of Science in Zoology from Iowa State University and a PhD in Anatomy from the University of Minnesota Medical School. She did a postdoctoral training at the University of California, Irvine, and at the Karolinska Institute. She was on the faculty of the University of Miami School of Medicine until 1994, when she began her work with nonprofit organizations, including the Tuberous Sclerosis Alliance, Genetic Alliance, Citizens United for Research in Epilepsy, and the United Coalition for Health Professional Education and Genetics. Dr. Whittemore joined the staff of the National Institute of Neurological Disorders and Stroke at the National Institutes of Health in August 2011. In this role at NIH, she oversees a grant portfolio focused on the underlying mechanisms of the epilepsies, including the genetic causes, animal models, translational and clinical epilepsy studies, and sudden unexplained death in epilepsy, as well as grants in MECFS. She is a well-known leader as a team in the Trans NIH Working Group, as well as in the MECFS Research Roadmap Project. Thank you so much, Vicky, for joining us today and for your leadership in bringing new researchers into the field of MECFS research.
Thank you very much, Beth. And thank you for the kind introduction and for inviting me to participate in this webinar today. So just briefly, I just wanted to inform those of you who may or may not have tuned in last week. So on Monday last week, we held a very successful one-day workshop on MECFS for young and early career investigators. So we had individuals all the way from someone who was a high school student, a couple of undergraduate students, some graduate students, postdocs, some junior faculty, and then we were joined in the afternoon by some individuals who are not so junior, but we were welcome to join them to our workshop. for a one-day meeting to really exchange ideas about the research that's going on, to network with one another, and to establish those collaborations between the young investigators that will carry them forward into careers in MECFS research. So there were several presentations that touched on the role of viruses and viral infections in MECFS, and a couple of individuals who spoke on long COVID as well. There were both oral presentations and posters. And so between the individuals in the room and the individuals who joined us by Zoom from around the world, there were about 100 young early career investigators, which is quite exciting to have that pipeline of new investigators in the field. So that following that one-day workshop, we had a two-day NIH MECFS conference that was organized and supported by NIAID, the National Institute of Allergy and Infectious Diseases, that brought together investigators to present on their updates and progress on MECFS research. Again, many of them touching on the role of viruses and viral persistence in MECFS and in long COVID. So it was, for us, I think, really an excellent conference that showed the progress that we've made since the last conference that we held in 2019, which I think has really brought us a long way toward understanding the underlying etiology of MECFS and really helping to shape and address the questions going forward, which is something that we're also addressing, as many of you know, in our strategic planning process to develop an MECFS research roadmap. So the video recordings from the conference last week will be available soon, and we'll send out an email announcement as well as post them on the NIH website when those are available. Today, it's my pleasure to serve as the moderator for the two speakers that have been invited to present their research. The first presenter is Dr. Bhupesh Prusty, who's a senior researcher and principal investigator at the Institute for Virology and Immunobiology at Julius Maximilians University in Würzburg, Germany. He earned his doctorate in biosciences from Jamia, if I'm saying this right, Millia Islamia in New Delhi, India. He was a postdoctoral fellow at the German Cancer Research Institute in Heidelberg, Germany, and his research is focused on innovative methods to understand molecular mechanisms of HHV6 and 7 latency and reactivation. He will join us today to give, well, before I give you that, I did want to add that Dr. Prusty was one of the inaugural recipients of the Solve MECFS Ramsey Award for a project on HHV6 mediated mitochondrial modulation and its association to MECFS. So with that, I'd like to introduce Dr. Prusty to give us his presentation on understanding infectious origin of MECFS through the recent pandemic. Welcome, Dr. Prusty.
Thank you, thank you very much. I guess
Dr. Prusty, you should be able to advance slides now.
I'm trying. It's not working.
We're still seeing that introductory slide.
So good afternoon, everyone. First of all, thank you for inviting me today to this webinar to discuss some of our recent work on MECFS. As you might know, my name is Bhupesh Prusty, and currently I'm located at Institute for Virology and Immunobiology in Germany.
Prusty, could you request remote control again, please? Give it just a second and try again, please. Try again, please.
It's too slow. Can I just click next?
Okay.
Seems to be. Yeah, okay. Okay, so as we know, MECFS is a systemic disease without any known biomarker, or even without clear knowledge of the mechanism of the disease development. One of the reasons behind this probably lies in the fact that majority of the patients are actually diagnosed very late with MECFS. And sometimes disease diagnosis takes years. And by this time, we mostly miss the very early stages of the disease development. During the late stages of the disease, we only see the chronic aspect of the disease with several varying clinical features from patient to patients. Today, I'm going to share some of the key findings from one of our recent papers, which is currently available in a medRxiv for your information and is under revision at this moment. This study actually includes roughly around 100 MECFS patients. And today I will also share some of the data, which is not the part of this preprint, but it includes also additional independent cohort of patients. We have studied different aspects of MECFS disease development in this paper, but today I'm just going to focus on only one aspect, and I'm going to compare them with what we studied in MECFS patients with healthy controls. We believe in infectious origin of MECFS. Hence, to understand the very early stages of MECFS development, we have compared our MECFS results with SARS-CoV-2 infected patients after 6 to 12 months of their first infection. This is very important to understand these results. And we have divided these patients into 3 different groups. The first group, we can call it as a no long COVID group. Basically, these patients had once the COVID-19 infection, and within 6 to 12 months, they have more or less recovered without any clinical symptoms. The other 2 groups include the mild long COVID group. We can also call it as mild to moderate long COVID group. And the last group is severe long COVID group. And these patients are separated, the different grades of long COVID is being separated by a scoring system, which was published by our own German colleagues in 2022, and also by the Recover study from USA in 2023. So the philosophy of my lab is based on one topic, which is my friend and the mentor, Bob Navio, postulated, and that's called the hyperactivated cell danger response in MECFS patients. What it means is that possibly only a very small number of cells in a human body is carrying the actual disease condition, whether it's infection or any other mechanism. It's not the whole body which is carrying the disease, rather only very few number of cells which are carrying the disease. And these few cells can actually communicate this threat to the other nearby healthy cells through this cell danger response. And thereby, this process amplifies the sickness response. Roffer also calls it as a mosaic disease. And when we say that only very few number of cells are actually infected or carrying the disease, it means that what we are searching is actually a needle in a haystack. So first, Ron Davis and then Øystein Fluge from Norway raised the idea of something in the serum, because serum from MECFS patients can actually recapitulate some of the aspects of the disease in healthy cells. We in our lab also focus on the same thing, and we also observed the same thing. My lab is focusing on finding these factors. I believe it is not a single factor. Rather, it is a combination of many factors. And one such factor that today I'm going to focus is immunoglobulins. When I say immunoglobulins, it doesn't simply mean the antibodies. Rather, It is the complete immune complex, including both the antibodies and the bound antigens to these antibodies. To understand this process, we have started purifying immunoglobulins from the patients and the control sera. We have established a biochemical pipeline for this, and we are building sort of an immunoglobulin biobank in our lab. What we do is we take these immunoglobulins and we do different type of experiments. So, for example, when we exposed immunoglobulins isolated from severe MECFS patients to healthy cells, we observed a dramatic collapse in the mitochondrial network. If you look into the figures on the left-hand side of the screen, you can see immunoglobulins isolated from a healthy individuals exposed to primary human endothelial cells, and on the right side, immunoglobulins isolated from a severe MECFS patients. And you can see that the mitochondrial network, which is basically green and yellow in color, is collapsing and coming to the periphery of the nucleus. We not only quantify this using confocal microscopy and looking into average mitochondrial surface area, we could also show by Western blotting, for example, on the right side, that when we expose immunoglobulins from the CFS patients, we see decrease in mitofusin 1 and mito-PLD or PLD6, which are the proteins which actually keep the mitochondria fused and elongated. So these results suggest that the immunoglobulins from severe MECFS patients are capable of changing the architecture of the mitochondria in the target cells. Sorry. So the obvious question which is coming from this is, what is there or what is not there in these immunoglobulins that is causing this mitochondrial phenotype? To understand this, we are carrying out mass spectrometry analysis of these isolated immunoglobulins to not only to see what portion of IgG and IgM is there in it, also to understand what are the different proteins which are there in it. In this type of study, we see some of the proteins which are present in a higher amount within the immune complex of MECFS patients. And some of the proteins are also significantly less within these immune complexes. Here is one example. One of the protein called ficolin-2 or FCN2, which is present in a higher amount within the immune complex of MECFS patients. Not all MECFS patients, but quite a substantial proportion of MECFS patients. We verified this, so you can see here that one of the dots over here is FCN2, and this is one of the patient versus one of the control. Here in the middle figure, you can see that when we combine all the healthy control versus all the MECFS patients, we see that MECFS patients on average have high amount of FCN2. So we verified this data using ELISA and found that FCN2 levels are high in a subset of MECFS patients. On the right side graph, you can see. However, when we compared some other autoimmune diseases, for example, sclerosis patients, we also saw that quite a large number of these patients also have high amount of FCN2 or higher amount of FCN2 in their serum. So probably this increase in FCN2 or ficolin-2 is just because of the autoimmune nature of MECFS. Now coming to the next slide, as I mentioned, there are only very few proteins which were actually increased in the within the immune complex of these patients. At the same time, some of the proteins were also decreased, and three of the proteins which attracted our attention is transferrin or serotransferrin, alpha-2 microglobulin, and fibronectin. So in this heat map, you can see that healthy controls normally have higher amount of these proteins within the immune complex, and not all again, but majority of the MECFS patients that we have studied so far had lower amount of these proteins within the immune complex. Today, I'm going to focus on only one of the protein that is called fibronectin or FN1. The mass spec study showed that fibronectin is decreased to different degree within the immune complex of these MECFS patients. Now, this raises question, is there an overall decrease in fibronectin amount in the blood? That's one possibility. Or there is a decrease in the antibodies that can bind to the fibronectin. Just to let you know that fibronectin binds to C1 antibodies and C3 antibodies and other antibodies. So we checked both IgG and IgM against fibronectin in our purified immunoglobulins by Western blotting. On the lower panel, you can see the Western blots. So just a representative Western blot here showing healthy control versus severe MECFS patients. And you can see that there is actually no change in the amounts of IgM or IgG against fibronectin. So this means that potentially there is some sort of translational modifications. This is again a hypothesis from our side that possibly some post-translational modifications are going on within fibronectin, and that post-translational modifications are preventing the fibronectin to bound to these active immune complexes. Now, next we decided to measure the fibronectin amounts in the blood. Now, here comes the question where to measure the fibronectin. Now, we can measure in plasma as well as we can measure in serum. Now, I'm bringing this topic over here because this is very important to understand what we are measuring and what is the function of fibronectin in serum and in plasma. There is a basic difference in amounts of fibronectin in plasma versus serum. As fibronectin is part of the blood clot, the amount of fibronectin in serum only represent the circulating fibronectin that is not incorporated into the blood clot. So when we prepare the serum, we let the blood clot. So a part of the fibronectin goes into the blood clotting process. What we isolate is the free circulating fibronectin. But when we talk about plasma fibronectin, that is actually includes both clottable fibronectin as well as the free fibronectin. This means that plasma fibronectin amount are always higher than the serum fibronectin amounts. So we first took a independent cohort of samples from Sweden, where we compared 40 healthy individuals versus 40 MECFS patients. And we took the serum and plasma isolated at the same time, and we tried to see what we see in serum versus plasma. Each dot in this figure or in this graph represents fibronectin amount from one individual. As I mentioned, as expected, the plasma fibronectin in healthy controls as well as in MECFS patients was substantially high in comparison to the serum amounts. However, the plasma fibronectin amounts in MECFS patients particularly was highly variable. Interestingly, the serum fibronectin was significantly higher in MECFS patients. So you can compare the HC means healthy control serum to the MECFS serum. We see a significant increase in the fibronectin amounts in the serum. However, when we do the same in plasma, healthy control plasma versus MECFS plasma, even though the trend is there, the statistical significance goes away. Now, this data is very important for us, and it's also corroborating the data which is already known in the field of MECFS, that probably there is a clotting defect is there in MECFS patient blood, and this results strengthen the previous observation in this regard. Now, this data is based on a fully independent cohort of samples from Sweden. So the next step was to look or to check the results in other cohorts of samples. For example, here, what I'm presenting is our own cohort of samples from Germany. And if we compare the healthy controls versus MECFS patients, once again, we see a reproducible data where MECFS patients have a high level of circulating fibronectin compared to healthy controls. Once again, henceforth, all the fibronectin studies will be done in serum, not in the plasma, as we see the results in the serum. Yeah. So when we divided this total group of healthy control versus MECFS into a male and female gender, we observed a significant difference in fibronectin amounts in the male. And this is simply because males have normally low amounts of fibronectin in serum in comparison to the female. We also found that the severe patients, severe MECFS patients with a very low quality of life have very high amounts of circulating fibronectin in the serum. Once again, coming back to the infection-induced MECFS theory, we all know long COVID patients have a great deal of similarity to MECFS. Even though there are differences in disease development process, some of the critical features of MECFS overlaps with long COVID. Hence, we studied the serum fibronectin amounts in our COVID-19 cohort of patients. We found that increased amounts of fibronectin in the serum of severe long COVID patients within the first 12 months of SARS-CoV-2 infection. When we investigated some of the patients, these no long COVID, mild long COVID, and severe long COVID patients, we did a follow-up cohort and picked some of the patients after 18 to 24 months of first COVID infection. We saw that the fibronectin amounts are even increased in these patients. And as usual, we compared these results with the sclerosis patients, just to divided them into groups, sclerosis patients without fatigue and with fatigue, where we did not see any increase in the serum fibronectin amounts. During this time, we had an opportunity to study around 20 MECFS patients who had actually developed MECFS post-SARS-CoV-2 infection. So let's call them as a PCS MECFS group. These patients saw the maximum increase in serum fibronectin amounts. If we separate them into male and female groups, we can also see that both male and female, they saw increase in the serum fibronectin amounts. And I predict that if we measure serum fibronectin in these patients after another 3 to 5 years, like we do in the severe MECFS patients, we probably will not see much difference in the amount of fibronectin in the serum because the body has been adapted to these changes over the period of time. Hence, the early time of the disease development is possibly the best time to identify these markers and to understand their contribution to the disease development. Now, I will wrap up these results with 2 more slides discussing on the potential outcomes of this type of changes in the immunoglobulins. Now, one must be wondering what these immunoglobulins are actually doing. Are these immunoglobulins when we are exposing cells, healthy cells with immunoglobulins isolated from patients, are they entering into the cells and doing something? Immunoglobulins rarely enter into the cells. However, they can bind to antigens and help in complement activation. And these activated complement proteins can then bind to the cell surface receptors, and they can regulate the physiology of the cells. However, recently, it was shown that antigen antibody binding is essential for the functioning of the many of the cells, particularly the B cells. Now, FC receptors on the surface of the cells, they can bind to the antigen antibody complex and thereby regulate the functioning of the B cells. And in the absence of the antigens, antibody may not function similarly. So it is known from the literature that different fibronectin components can actually have different roles on the mitochondrial architecture. So if you expose cells to a full-length fibronectin or fragmented fibronectin, depending on the type of fibronectin, they can cause mitochondrial fragmentation or mitochondrial fusion. So we predict that these IgM and IgG bound fibronectin, they have a pro-survival function. They help in better mitochondrial architecture. And when the fibronectin is not there with the antibodies, they might cause a mitochondrial fragmentation phenotype. We need more experimental evidence for this. However, this is one of the hypotheses. And one of the, I'm missing a slide over here. One of the recent paper which came out in the Cell showed that long COVID patients have a low amount of serotonin in these patients. And it's very perplexing to know that serotonin can actually be used to modify fibronectin. This process is called serotinylation of fibronectin. So it will be a future topic for us to understand if the serotonin, which is low in the long COVID patients, can actually be used by the fibronectin to modulate this fibronectin in a way that this is not incorporated anymore into the active immune complex as well as in the clotting process in these patients. So with this, I would like to thank my collaborators, my funders, and the entire patient community who support our work and all of your support and encouragement. And thank you very much. I will be happy to take questions now on or at the end of this webinar. Thank you.
Thank you, Dr. Prusty. We'll take questions at the end after Dr. Lee's presentation.
Thank you.
Thank you very much for that interesting presentation. So the next speaker is Dr. Dawei Lee, who's an associate professor at Texas Tech University Health Sciences Center in the School of Medicine in the Department of Immunology. He's a principal investigator for the Bioinformatics and Computational Genomics Lab and director for the planned Center for Genomic Medicine and Bioinformatics. He received a doctorate in genetics from Shanghai Jiao Tong University in China and has been trained in human genetics, genomics, and bioinformatics. He completed postdoctoral training at Rockefeller University and has been a research scientist at Yale and on the faculty at the University of Vermont. So his current research focuses on developing innovative genomic and omics research methods and applications to MECFS. In 2018, he was awarded the Solve MECFS Ramsey Award for endogenous retroviruses and their expression in chronic fatigue syndrome, which is what we're going to hear from Dr. Lee about today. So welcome, Dr. Lee.
Thank you. Thank you, Dr. Whittemore and Dr. Bremer and Dr. Unger for giving me the opportunity to present today. Next. Oh.
Yes.
Okay, it works. Thank you. So we just learned the virus may trigger MECFS, such as Epstein-Barr virus and SARS-CoV-2. Most people have these viruses, but not everyone has MECFS. So I suspect endogenous retrovirus, briefly, ERV, is a key factor here. Possibly, only people with certain genetic predisposed, such as with certain ERV variants or ERV transcriptional activation, can develop a chronic disease after acute infection. Today, I will introduce the possible link between MECFS and between the ERV and MECFS. So what is ERV? ERV are derived from ancient retroviral infection and the virus integration into our ancestors' germ cell several million years ago. ERVs are now fixed and part of our human genome. We insert it in the ERV inserted in our genome in every single cell we inherited from parents into every single cell. The protein coding genes account about 1.5% of human genome. By comparison, ERVs account for 8%. Each human genome has about 20,000 protein coding genes. By comparison, ERV genome has at least about 600,000 ERVs, which that is about 30 times more than protein coding genes. So this ERVs has various of functions in human health. For example, some ERVs can produce viral proteins or non-coding RNAs. Some ERV can fuse with nearby genes to make fusion transcripts. ERVs has promoters and enhancers and can upregulate the expression of nearby genes. Methylated ERVs can silence downstream gene expression. Because of ERVs are repeat sequences, ERV can facilitate homologous recombination leading to copy number variations. Because ERVs has inserted in the human genome for several million years, so the human genome has co-opted with ERVs for various benefits. Here is one example. In early embryo development, the placenta needs ERV proteins for its cell-to-cell fusion of the placenta. ERVs also regulate the key hormone gene important for pregnancy. So some ERVs ERV proteins mediate cell-to-cell transfer of mitochondria, as shown in red color in this slide. We can imagine if this ERV protein is dysregulated, the cells will not be able to transfer mitochondria fast enough from a source tissue to the destination, such as muscle. So we know ATP production is wrong in MECFS. Research is needed to examine if ERVs are related to MECF antigen production. ERVs is also the only biological material that is derived from virus and also can pass from parents to children in Mendelian inheritance model. ERVs are typically silenced by methylation, but ERVs are viruses by nature, so they can be reactivated to produce viral products. ERVs does not need to make a live virus or viral protein. Viral RNA is sufficient to trigger immune response because once activated, ERVs can make double-strand RNA, which will be recognized by the human cell double-strand RNA sensor as foreign RNA, which then trigger type I interferon, leading to the inflammatory immune response we observed in MECFS. ERV also has a hormone-responsive elements adjacent to the enhancer and promoter. You can see here. So ERV activation can be additionally enhanced by female sex hormones. So we know 3 out of 4 MECF patients are women. So to study the MECFS, ERV MECFS link, the first question is how to detect ERVs in MECF patients. So ERV detection is very challenging. For the ERV variant, the ERV variants are not captured by SMP genotyping rate and not in the GWAS imputation database, not in standard and next-gen sequencing pipeline. And the ERV expression is not in the standard RNA-seq pipeline or the RNA-seq RAP-seq database. Existing ERV detection methods are very limited. So we developed a novel method we call the ERV caller to use the next generation sequencing to genome detection and genotype each individual ERV on the genome-wide level. ERV caller can accurately detect and genotype each individual ERV using raw sequencing data and achieving about 97% of accuracy based on our simulation data. We use the ERV caller into the various of existing genomic sequencing data, including the 1,000 genome samples. Our methods identify novel ERVs that cannot be identified by other approaches. We did the PCR and Sanger sequencing to validate those novel ERVs we identified. All the genotypes were verified by Sanger sequencing. This figure shows just 2 randomly selected samples. In short, our method accuracy is very high. So our ERV caller, the platform process each sample one by one and generate a standard format of genotype data that are ready for genome-wide solution study in any human samples. So we used this platform to analyze our existing MECFS sequencing data. We first used the ERV caller to analyze whole genome sequencing data from 20 severely ill MECFS patients. We found many distinct individual ERV variants. Here I only show 8 of them. This palette data is the patient only without controls. So I compare the patient's blood measures by ERV genotypes. As you can see, for example, here is 1 copy, 0 copy. This group of patients have no ERV. These have 1 copy, they have 2 copy. Yeah. Right. So as you can see here, the genotypes of the ERVs are associated with several cytokines or blood measures. Some of the cytokines were reported to be important to MECFS in the literature. For example, Dr. Mark Davis' group found the top 3 here are positively associated, correlated with MECFS severity. we found the same three are also positively associated with ERV genotypes in the same direction. So we also analyzed our existing RNA-seq data from MECFS case control samples. In this figure, I use the differentially expressed ERVs to do a principal component analysis. As you can see here, the patients are clearly separated from controls. The blue here is the patients. We also found a number of ERVs uniquely expressed only in patients but not in controls. Use of this MECFS specific ERVs or a subset of them can distinguish MECFS versus the healthy status of our examined samples. So I also analyzed the public available RNA-seq data of MECFS versus control before and after CPAD exercise. We found a number of differentially expressed ERVs. Here, I only show one example, as you can on the right side. In healthy controls, this ERV expression was low. In the day one baseline, its expression went up with the first exercise and then came down to baseline after a few days of rest. By comparison, in the MECFS, the day one baseline of this ERV is already high and did not come down. We found a lot of differentially expressed ERVs. Those ERVs are novel. Nobody has annotated them before, so we don't know their exact functions. So I checked what are the genes co-expressed with these ERVs. So our analysis produced interesting networks. Here I randomly select a small part of the network here. So the ERVs, as shown in yellow color, are well connected and centered in the network. When I check the most connected genes, many of the genes are related to retroviral activation or mitochondrial functions. The network appeared not at random. So we are currently examining the question: does ERV drive genes, or the genes drive ERVs? So our samples are mostly from Americans. Our European colleague, Dr. Elisa Ultra's group, analyzed the ERVs in Europeans. So they got basically the same results as ours. They use the microarray to quantify ERV expression in MECFS, fibromyalgia, and the patients with both. They also use the differentially expressed ERVs to do a principal component analysis, as shown here. The ERVs clearly separate MECFS from controls, also from the fibromyalgia. Interestingly, the same procedure was done by using protein genes, but as you can see, only ERVs can distinguish MECFS. Similarly, they also examined the genes correlated with ERVs. They found several modules, among which the most significant module, the genes are involved in the T cell activation, and the co-expressed ERVs are low in controls and fibromyalgia, but high in MECFS. In short, the microarray data in Europeans are pretty consistent with our genomic sequencing study in Americans. So a healthy normal cell should only have very low basal ERV expression. Long COVID mirrors the major symptoms of MECFS. A recent literature showed the SARS-CoV-2 virus can activate ERV, the expression of many ERVs. By comparison, other respiratory viruses like flu can only activate immune-responsive genes, but cannot activate the ERVs. So COVID can cause long COVID, but flu does not lead to long flu. For many years, we've known Epstein-Barr virus can also activate ERVs. We know after verification, we first see IgM in early stage and then IgG in the later stage. This paper found increased ERV IgM in COVID patients, but in the MECFS patients, the ERV's IgG increased. So this is consistent with our anticipated progression from COVID, long COVID to MECFS. So this paper also concluded long COVID can reactivate ERV expression regardless of if you have COVID symptoms or not. So the ANH MECFS conference held last week had a lot of great talks, which together show abnormalities in almost all aspects shown in this figure, such as the brain neuroinflammation, microbiome, metabolism, proteins, gene expression, epigenetic, etc. Some of the abnormalities may eventually lead to a diagnosis, biomarker, or even treatment. I hope our future research along the ERV to MECFS link will add more data into the causal mechanisms of this disease. Only if we know the cause, we can better treat, reverse, even prevent the disease. I thank patients and their caregivers for donating their research samples. The samples used in this talk were from Dr. Ron Davis, Dr. Shell's groups, and Dr. Hansen's groups. Our research is supported by the South MECFS Ramsey Award, Open Medicine Foundation Patent Award, and the NH R21. Thank you very much.
Thank you, Dr. Lee. I'll start with a question for you. So what makes your ERV detection methodology and your research different than what's come before, than previous research?
Thank you, Dr. Whittemore, for the great question. So in the past, the ERV detection was mostly based on the probes, such as PCR. So if we design a PCR probe, we will actually capture a lot of ERVs. There will be a pool because although each human genome has about more than 600,000 of ERVs, but those ERVs share a lot of sequence similarities. So a probe is typically will capture a lot of ERVs. So we will see in the past research, mostly they are studying a pool of the ERVs. But I suspect those ERVs are different. Although they are sequencing, they have similarities, but they are on a different chromosome. Think about the chromatin three-dimensional structure. They may have very different functions. So in our research, we use the next generation sequencing to do a non-hypothesis-driven screening. So our bioinformatics approach, we are trying to distinguish each individual ERVs. So we will find the resolution on the chromosome, nucleotide resolution. We know this ERV is in this chromosome from database from nucleotide 1000 to 2000, for example. So that's what that make a huge difference regarding the. So we are trying to separate each of the 500,000 ERVs one by one. Thank you.
Yeah, thank you very much for that explanation. Dr. Prusty, if you could turn your camera, and I have a question for you. So at the very end of your talk, you talked about sertraline, and I'm just interested if you would say a little bit more about how fibronectin levels are regulated and the role of sertraline. If you could just give us a little bit more explanation of that.
Yeah, I had a slide. I don't know exactly why the slide is not there, but anyway, fibronectin is an essential component of the extracellular matrix, and it's a huge 420 kilodalton, 210 kilodalton protein, and present in the form of heterodimer. And it undergoes many different type of modifications. And one of the modification which seems to be very interesting is the serotoninylation, is the sense that, so normally serotonin is produced in a limited amount in the body, and it has its own function as a messenger controlling a different type of physiological activity. But it is interesting to see that serotonin under specific conditions yet unknown is recruited directly to the fibronectin protein. It binds to fibronectin and makes the fibronectin serotonin-related fibronectin. Now, it's just a hypothesis at this moment is that the paper which came out in the Cell that the serotonin is decreased in these patients. And what we see is upregulation of the circulating fibronectin in the patients. It just makes hypothetical understanding at this stage is that probably the serotonin is recruited. We have some experimental evidence that yes, it is possible, but we need more data for this. What happened is that when fibronectin is serotonin-related, it is serotonin-related in sites, specific sites, which might inhibit its function as a antigen because the antibody binds to that site. And by these changes, we believe that the antibody binding site is not available anymore to the protein. Yeah, but again, we need more data for experimental data to support this.
Yeah, thank you very much. That's very interesting. Yeah. So at this point, I'll turn it over to Christine to moderate the Q&A.
Thanks so much, Vicky. So just a reminder, everybody, we'll do the Q&A now for the general Q&A portion. There are 3 ways to ask a question. If you're joining on Zoom, you can raise your hand by clicking the raise hand button under the webinar controls at the bottom of the screen, or you can type your question in the Q&A chat box. That's being monitored by one of our team members here. If you're joining by phone only, you can enter *9 on your phone to join the question queue. And when we announce it's your turn, you can press *6 to unmute yourself. So I think let me start here. So the first question that I have is a question for Dr. Prusty. It says patients with Ehlers-Danlos or joint hypermobility frequently have comorbid POTS, MECFS. Do you see a link with the work you are doing on fibronectin, elastin?
Yeah.
Sorry.
Yeah. Yes, I saw the question. Yeah. Thank you. We have not tested the EDS patients at this moment. We do not have any clinical collaborations where we can get these patients. But during the last 6 months, we have been so basically we have been asked by the reviewers to validate our results in a completely independent cohort of patients, and we have been doing it going into multiple different centers within Europe, and having well characterized cohort of patients and trying to see it. And we have started to look into sclerosis and some other cancer patients with fatigue and things like that. But we have never got an opportunity to look into the EDS patients. We are open to all sort of collaborations and looking into other type of disease conditions and to compare it if this is a case. But one thing I would like to mention it here. Today's talk was only 15 minutes, and I could not cover the other part of the talk where we look into the IgM against fibronectin. And this seems to be very interesting that the IgM against fibronectin is capable of differentiating infection-induced MECFS from other type of MECFS triggers like trauma, stress, or the cranial nerve related MECFS, which is completely non-infectious trigger MECFS. It is capable of differentiating, which is very interesting for us. And this also brings us to the point that anything which is probably an infectious triggers, including the COVID-induced MECFS, is probably fitting more to the fibronectin hypothesis rather than any other trigger of MECFS. Yeah, thank you.
Thanks so much, and sorry for flubbing that. I'm a communicator, not a scientist. The next question that we have is, how is CDC getting word out to the medical community and healthcare data systems about the new ICD code for MECFS? Dr. Unger, would you like to take that one?
Sure.
So sorry. Okay, where were we? Oh, the question.
Yeah.
Yes. So we have, I think we mentioned the new code in our last C call. It was very new at that point, and we have included the information on our website, and we will be including it in all of our educational materials. We think that it is really important to get the word out, and we'll do what we can to think of other ways to get it out.
Okay, great, thank you. So the next one is for Dr. Lee. It says, I'm a longtime person with ME. I thought it was triggered by a flu. Now you are saying flu doesn't trigger ME. Was it perhaps triggered by an EBV?
Thank you for the question. So I think for now, there is no evidence show that flu can trigger ME. And also, I think that the data is also not sufficient to prove the EBV can really is a cause or trigger. But I personally think that there will be a very wide range of triggers, the factors that can trigger the MECFS. But for now, we don't know yet what they are. But SARS-CoV-2, it looks like it's one of the triggers. But what are the others before the COVID pandemic? We don't know yet. That's what we are trying to find out.
Great, thank you so much. So the other one the next one is not directed to a specific person. So if either of our guests would like to respond, or both of you, that'd be great. The question is, are any of your findings pointing towards specific treatments or experimental trials?
Yeah, so I can answer this question. The results at this moment suggest that just in the form of the fibronectin, it's a protein which is produced in response to inflammation. We have some initial evidence from our collaborators in the US that fibronectin is also produced in response to DUTPase, particularly from EBV. So we believe that any treatment conditions which takes care of the infection probably will also take care of the increase in fibronectin. As such, at this moment, nothing is known about the treatment for high fibronectin amounts, but we have to keep in mind that fibronectin is also essential for the normal healthy body. So I guess the root cause of the increase in fibronectin probably should be taken care of in terms of treatment rather than increase in fibronectin. Yep.
Great, thank you. Dr. Lee, did you want to answer that question as well?
So
I can repeat it if you'd like.
Yeah, so regarding the possible treatment, so I guess like for example, if the ERV MECFS link is proven, then it's possible we might be able to repurpose FDA approved generic antiretroviral or anti-inflammatory drugs to reverse ERV effects. So I learned of, and also we know I think there is already ongoing the antiretroviral clinical trials to suppress ERVs in multiple sclerosis and also in ALS. Maybe we could use some time to test this in MECFS as well. I think probably we should. And also I learned from several patients. I learned from them, they self-used some of the antiretroviral drugs, and they see they have improved the symptoms. The second is about I saw from literature about epigenetic nutrients. So the MECFS patients use the epigenetic nutrients actually improve the symptom improved regardless of the deficiency of status of this. Yeah, thank you.
Okay, thank you.
By the way, I also want to mention that regarding the specific for the ERVs, I see from literature, the ERVs also currently is emerging targets for the immunotherapies, which a lot of in the cancer treatment. But because ERVs are different, if we find the ERVs in the MECFS, there could be, so immunotherapy could be one of the approach in the future down the road.
All right, excellent, thank you. So the next question is about ICD codes. It is there a way to encourage long COVID clinics to screen their patients for MECFS? And if they qualify for MECFS diagnosis, as many will, also put in the code for MECFS in their record. Would you like to take that, Dr. Unger?
Sure, I can start at least. It's a really good question. We have included information about MECFS in our long COVID and recovery illness program. And I think that it's pretty clear that it's very difficult to talk about long COVID without mentioning the similarities to MECFS. So we do encourage the physicians to identify MECFS in their patients through their diagnostic workup and to be aware of the similarities. The post-COVID condition code also encourages physicians to diagnose other conditions that are identified in these patients and lists MECFS, I believe, as an example. But CDC does not direct anybody how to code things. That really is up to the clinicians. We do point out that it will be much easier for us to track the illness in the population if people use the codes correctly.
Great, thank you. So the next question is for you, Dr. Unger, which is CDC released their new prevalence survey, which inflated the number of people with ME. Why did CDC use a telephone survey asking people about tiredness and fatigue rather than assessing people with viral and biomedical evidence to determine the prevalence? And why did CDC not use experts criteria, MEICC, which would capture the accurate prevalence?
Thank you. We really wanted to get a national estimate of MECFS, and the only way to do that practically was through the National Health Interview Survey. Now, the National Health Interview Survey does not ask how the healthcare provider made the diagnosis, so we don't know what method was used or what case definition was used. And we do know that there are limits in using a self-provided report of a physician diagnosis. Nonetheless, it's a baseline for us to evaluate and make other important health connections through the National Health Interview Survey questionnaires. And the Canadian National Health Service used a very similar approach.
Right, thank you. So the next question I have is, one clinician researcher reported not diagnosing MECFS in long COVID cohorts when it was present because certain research studies would not accept those patients. Any ideas on how to address that?
That is I had not heard that as a problem. And I think, you know, study protocols have their own rules. And I don't think there's a way for CDC to address that. I don't know if anybody else would comment. Sorry.
Okay. So the next question is for Dr. Prusty. It says, if clotting defect tends to show up early in ME, but but if the body adjusts later, does that mean clotting issues are unlikely to show up later in disease progression?
Yeah, good question. No, it's not the case. What I meant that the body adjusted the amount of the fibronectin particularly is that over a period of several months to years, when, as I mentioned, the fibronectin is a marker for inflammation. Over the period of time when other type of chronic conditions appear and patients go on different type of treatments and things like that. So the increase in fibronectin, what we see in the initial period goes down, but that doesn't mean that all the functions of fibronectin, whether it's a defect in clotting process or it's incorporation to the immune complex changes. It's just that the sudden increase in the fibronectin that we see just after the infection, like in COVID-induced MECFS, is diluted over the period of years. It is increased over a period of time, like I said, within 8 to 12 months, it was increased, but it increased even further with 18 to 24 months. But as we see in MECFS patients, most of the times patients are sick for several years, sometimes 20 years, 30 years. So it's very hard to see the similar significance in those patients. Yeah.
Okay. So the next question is, when are we going to see clinical standards for long COVID clinics? So they might begin properly diagnosing their ME patients and also finally stop them from harming patients, diagnosed or not, with outdated anti-science therapies like GET graded exercise therapy.
Well, as you probably know, HHS has established and within the Office of the Associate
Secretary.
Yes, Associate Secretary for Health. And they have issued reports. I don't think they're ready to give a national mandate or guide to diagnosing long COVID, but they certainly are making sure that information about the reality of the illness and the importance of understanding the impact that exercise can have through post-exertional malaise.
Does the CDC have plans to help mandate or at least encourage and publicize use of the new Mayo Clinic proceedings guidelines for managing and treating ME?
Well, that will be a good reference that I don't think we currently have it in our references for healthcare providers because it's new, but that is one that we can include when the new web pages are up. That would be the best way for us to refer to it.
Folks, I'm scrolling. Are there, okay, so Eileen, I think your hand is raised. I'm sorry, I've got multiple screens up here, but go ahead and unmute yourself or try, and then you can ask your question.
Hi, this is Eileen Holderman. Can you hear me?
Yes.
Well, first of all, thank you so much for this presentation. And also, I submitted a question in the Q&A. And I thank you so much, Dr. Unger, for answering that. I have a quick follow-up regarding that survey. And Dr. Unger, you and I worked so many years on the CDC website. And I'm gratified to hear that you're going to be making more improvements to it. But the recent press that centered around the survey's release had me very concerned because it was recycling old myths, debunked information about ME, buzzwords like patients report, the word fatigue, the word tiredness. We didn't hear anything about viruses and infectious disease as our esteemed presenters, Drs. Prusty and Lee, talked about. Can you talk to us a little bit about CDC's interactions with the press regarding all of that very bad press?
Well, let me, I'll start by saying, we did do an interview about it. We were asked just a very few questions, was really very brief. And then we don't have a lot of control over how it gets promulgated. And I'll turn it over to Christine, who helps coordinate this.
Sure, yeah, so I'll just mention that, Beth is right, we did not actually get media queries. Most of the reporters who are covering this just wrote from the report. And, we've been trying to, when we did talk to the one or two reporters, I can't remember, it was very, sort of like, what does this mean? And what can you tell us? But they didn't have a lot of specific questions for us. And then, as far as once we talk to them, we don't have any control over what their headline is or even how they cast it. We tried to give caveats when we did the interviews about what the study meant and what it doesn't mean, and how the numbers and things are different than you might see if you were looking at a population of people who go to some of the specialty clinics with the experts, or if you or if you did the survey a different way. And so, we tried to provide sort of the benefits and drawbacks from doing a survey that's just a national survey like that. But again, I mean, I used to be a newspaper reporter, and I will tell you that, a lot of times they're given very little space and not a whole lot of time or background on it. And
yeah, and I will say I was disappointed often that I saw chronic fatigue, MECFS, just abbreviated as chronic fatigue. It just pained me every time I saw that. So I knew the community would be hurt by that. And so the good news is at least it got some attention. And I hope that this will keep the interest up in this very, very significant problem.
And, I will just say, as someone who has been working on communications for MECFS for about 15 years here, that is probably the most that any reporters have paid attention. So I'm hopeful that potentially, if other reporters see it, we can keep up the good fight of trying to use the right words and explain to them about it. And hopefully see some more interest. In addition, now that long COVID is out there, that is, I think, also drawing people's interest. So I'm hopeful that those things will help keep it in people's minds. Okay, let me see. Hold on a second. I was talking, so let me see if I can scroll down and see if there are additional questions.
Ed Young had a very, very thoughtful commentary recently. He's a reporter for The Atlantic, and he did the commentary, I think, in the New York Times, and he really understands. And so there are reporters that hear it and learn from that. So we just hope that there's going to be more and more of them.
Okay. So the next question says, wouldn't more specified press releases be acceptable by the media to target the important points of MECFS? So I will say that most reporters get probably hundreds of press releases in their inbox every week. And it is incredibly hard to try to get them to be interested. One thing that we have done that has, I think, sometimes been helpful is to reach out to specific reporters who have been interested in the past, or to try to reach reporters like, say, healthcare reporters, or other sort of targeted ones. A lot of reporters basically are generalists who may write about, city planning commission one day and education the next and MECFS the following. And I think that is something that we have difficulty dealing with, because they are generalists, so they don't really necessarily know a lot about well, really any of the things that they're writing about. And that's really the best we can do, is just try to educate them when we talk to them. Oh, and then the next question I see is, where can I get a written transcript of this presentation? So we will get a transcript, and we will be getting it cleaned up, and we'll get it posted on our website just as soon as possible. If you go to the website, which is www.cdc.gov/ME-CFS, and then go under the section that says meeting, then you'll be able to find it there. All right. So this one looks a little bit more like a comment, and it says your explanation for media using incorrect terminology underscores the need to change the terminology on your end. Fatigue is an inappropriate word. Use of scientific language. Only please respectfully, you have the power to change the language for our community and can command great change for ME. So thank you for that note. I will say that, as I mentioned, we are working on revising and updating our webpage, and we will, we'll listen to any and all input that we receive, and we'll work on assessing it.
Right. And there was a comment before, and Vicky and I discussed this prior to the call during the NIH conference, a suggestion that we move away from the term MECFS, which HHS as a whole has adopted, and move strictly to ME. And that's a decision that the government as a whole has to make through the agencies. So the naming convention will move together. But thank you.
All right, and we're closing in on time, so this will be the last question. It says, will previous information about CFSAC meetings still be available in the archive website? Please retain this information. It's important in the historical record. Thank you. Yes, absolutely. We will not be taking anything that's currently in the archive site out of it, and we will be archiving any old meetings that are not going to be moved over to the new site. And then the plan is that, as we have these additional seat calls, basically the oldest one will roll off and the new one will be posted, but they will always be available. And we'll also post a link that has the how to find that stuff in the archives, so that everybody can find it if they need it. All right, well, thank you so much, everyone, for joining us today. We thank you to our speakers and to Dr. Whittemore for your help, and to Dr. Unger for your remarks. We will be posting a transcript and slides and a video just as soon as we can get them prepared. Thanks, everyone. Have a great day.
Thank you.
Thank you.